皆様、LINK-J のライブウェビナーにご参加いただき誠にありがとうございます。本日は LINK-J &UC サンディエゴジョイントウェビナーシリーズ第15回 with 京都ユニバーシティ、細胞内シグナル伝達と脂質研究セッション4、脂質膜研究に関する最新の手法をお送りいたします。私は LINK-J の高橋と申します。本日は画面でお示ししているプログラムに沿って進めてまいります。本日は日本の早い時間にもかかわらず100名を超える皆様からの事前登録をいただいています。アカデミア、製薬企業の方々が多いようです。このことからも本ウェビナーのトピックの関心の高さが伺えます。本日も UCSD と神戸大学の最新研究をお楽しみください。さて、本日の各発表に関しますご質問は Zoom の Q&A 機能からお願いをいたします。時間の都合上、すべての質問にお答えできかねる場合がございます。また、本イベントでは、ウェビナーの同時通訳機能、英語字幕をご利用いただけます。字幕は市販されているアプリをしており、LINK-J、UC サンディエゴ、神戸大学は語訳、訳文の欠落など、字幕に関する一切の責任を負いかねますので、どうぞご了承ください。なお、本ウェビナーの録画、録音、画面のキャプチャーは固くお断りをしております。最後になりますけれども、ウェビナー終了後に表示されるリンクより、アンケートフォームにお進みいただき、皆様のご意見、ご感想をお聞かせください。皆様に役立つセミナーを開催をしていきたく、ご協力をお願いいたします。それでは、UC サンディエゴ国際アウトリーチシニアダイレクター和賀さんより、イントロダクションとモデレーターのご紹介をお願いします。和賀さん、よろしくお願いします。
はい、おはようございます。Good morning, Japan, and good evening, San Diego. Hi, my name is Miwako Waga. I'm a Senior Director for International Innovation Outreach at UC San Diego. And it is really a great pleasure to continue this wonderful collaboration with Kobe University. And also, I'm so grateful for LINK-J and also the wonderful translators to make this very special event possible. I do hope that the audience will enjoy and appreciate the depths of this conversation, scientific discoveries and discussions, and appreciate it very much. Hopefully, they will reach out to Kobe University and UC San Diego for collaborations in research and education. So without further ado, I'd like to invite Professor Shirai of Kobe University to say a few words and also introduce the moderator from UC San Diego.
Thank you, Miwako. Hi, my name is Yasuhito Shirai. I am a dean and a professor at the Graduate School of Agricultural Science at the Kobe University. So, this is the 15th LINK-J and UCSD joint webinar, also the fourth session of cell signaling and lipid sciences. So we started this series of cell signaling and lipid science about 3 years ago. And I'm very pleased to tell you this. Based on this project, we have succeeded in getting the big grant, core-to-core grant from JSPS. So I think we can strengthen this relationship between UCSD and Kobe University. Today, we focus on the novel method for the lipid membranes. So we're going to have two professors from Kobe University and UCSD. From Kobe University, we're going to have Dr. Kenichi Morigaki, and from UCSD, we're going to have Dr. Christopher Obara. So I'm very pleased to introduce Dr. Kenichi Morigaki from Kobe University. Kenichi, please.
Hi, I'm Kenichi Morigaki. I'm going to tell about our research in the second half of this session. So I'm very excited to have this opportunity. Thank you very much.
I'm sorry, I made a mistake. I'm sorry. I have to introduce to Itai. I'm so sorry. Itai, please.
No problem. Thank you, Yasu. Good morning, everyone. Ohayo gozaimasu. It's really my pleasure to co-host this webinar with Professor Shirai. So as Dr. Shirai introduced, our focus today is going to be on new development of new methodologies to study lipid sciences and cell membranes. And so I'm going to introduce our first speaker, who is Dr. Christopher Ibarra from UC San Diego. So Christopher Ibarra has an appointment in my department, which is the Department of Chemistry and Biochemistry, as well as the Department of Pharmacology. And so he did his training at University of Florida in physics, and then his PhD at the National Institutes of Health in Maryland, before doing postdoctoral work with Jennifer Lippincott-Schwartz. And during his postdoc work, Dr. Ibarra made really seminal contributions to understanding organelles within cells using new microscopy techniques. And he has started his own research group here at UC San Diego within the past few months. And so he's continuing this research focus, and he'll describe some of the really exciting developments and approaches that they're working on. So without further ado, Chris, if you want to take over.
Thank you so much for the introduction, Itai, and thank you all for giving me a chance to speak. I'd like to tell you a little bit today about our ongoing efforts sort of at the interface of technology and biology to try to understand how individual proteins in membranes are coordinated to give rise to the biology that drives living cells. As I think most of you are probably familiar at this point, biology can happen on a lot of different spatial scales. And despite the fact that we've been studying that now for many years, I'd argue that we don't know a uniform amount about all of these scales. So for instance, many years of structural biology and cryo-EM now, and in the modern era, even computational techniques like AlphaFold have told us a lot about what individual proteins look like. And classic biochemistry has told us a lot about what those proteins interact with directly in living cells. Simultaneously, since the development of imaging technologies and fluorescent proteins, we've learned always a lot about where individual proteins go in cells, which organelles they're compartmentalized to, et cetera. But these two tools have an awful chasm between them, what we call the black gap in advanced imaging technologies, which is a space of spatial interactions that we really don't have good tools for bridging. And of course, electron microscopy has helped us now for many years to bridge this gap, but electron microscopy lacks the specificity to tell us where individual proteins are in cells. Instead, we can only see where structures are. And so over the last few years, we've seen an explosion of new technologies that are collectively known as super resolution microscopy and use light and optical techniques to try to help bridge this gap. And this is really the space where my lab is focused, which is that we are trying to understand how to take optical technologies and integrate them into these other things like structural biology and electron microscopy and conventional fluorescence microscopy to bridge across these gaps and try to understand how individual proteins give rise to biological phenotypes. So my preferred organelle to work in is the endoplasmic reticulum. You hopefully are familiar. This organelle is one continuous membrane network that makes up the nuclear envelope of all living eukaryotic cells and stretches out as a continuous convoluted network of membranes that are continuous all the way to the very edge of cells. And this organelle is involved in nearly every cellular process. It is the major site of cellular translation, the primary site of lipid synthesis and modulation, and amongst many other functions that it performs for living cells, it also maintains sites of specific contact with nearly every other organelle in the cell. And these sites are believed to be the primary drivers or controllers of the biology within those compartments. The organelle is also very dynamic. It undergoes structural rearrangements on several different timescales, both over seconds and also even over milliseconds. When I first began my postdoctoral work, we became interested in trying to understand something about the structure that underlies this network. One of the first observations that I made as a postdoc using super resolution technology was an attempt to try to understand what proportion of the endoplasmic reticulum was made up of curved membranes versus portions that were made up of flat membranes. And the approach we took for this was to take simultaneous low resolution images that are what people had been using before and couple these to super resolution technology that allowed us to see in higher resolution. But what we found, much to our dismay, was that structures that look continuous at one size or resolution often don't look continuous at other resolutions. And at the time, it was very confusing to us why this might happen. So we wondered if this was an artifact of fluorescently labeling proteins. Could these dark spaces that we saw on the edges of structures in the endoplasmic reticulum be the result of protein partitioning away from subdomains within the membrane? And to try to address this question, we went back and looked a lot at how their size and their dynamics. And what we realized was that these structures are actually too small to even see by traditional diffraction limited imaging. You need super resolution microscopy to resolve them at all. And their lifetime is in the millisecond range, which means most conventional microscopes will take an image that averages these out. And in an attempt to understand something about what the underlying structure for these were, we developed two technologies that we were very excited about, which directly image the underlying lipid structure inside the organelle. One of these is a single molecule technology known as lattice light sheet paint that individually localizes the position of single lipids inside of the organelles. And what you can see is this provides a very high resolution image of all of the membranes in the cell, but makes it clear that these structures we saw are not where proteins are partitioned, but instead actual holes inside the structure. There's no lipid inside of them either. And we could confirm this with a technology known as FIB-SEM, which is based upon high pressure freezing and freeze substituting cells and depositing osmium onto the membrane, and then doing 3D EM to try to understand what the underlying shape is. And hopefully what you can see here is that this very convoluted structure here, if we simulate what a light microscope would see, it looks like it's a continuous structure. So since we published this, there has now been a lot of work that has gone on by many different groups using many different technologies, not just the ones we introduced, which has identified that there are lots of structures in the endoplasmic reticulum membrane that are not visible by light microscopy because the details that make them up are too small. And so we recently put together a paper that tries to characterize some of these. And in this paper, we have this figure here, which I'm quite proud of, which is a series of 3D electron microscope reconstructions through 8 different structures that all happen within about 5 to 10 microns of one another in the close perinuclear region of the endoplasmic reticulum of a tissue culture cell. And so what's perhaps even more startling than how different these look is that everything from E to J, so the last 6 of these, are completely indistinguishable even by a theoretically perfect confocal microscopy image. And so what this led us to conclude was that if we want to understand where the ER is spread out in cells, light microscopy is a good tool for this because we can see the entire cell and we can see sort of general subregions of the ER. But if we want to understand what the underlying membrane structure itself is, we require 3-dimensional electron microscopy. And so I won't show you all of this because we've published it now in a series of steps, but the major conclusions that came from my postdoc work on this are that the large-scale distribution of the organelle is controlled by its ability to bind to cytoskeleton and other rigid components of cells, but the nanoscale structure, what we see in electron microscopy, is more likely to be controlled by the proteins that are sitting in the membrane at that site. And with this last observation sort of in mind, we ended up with a major question, which is if there are this many different structures inside of cells, membrane structures, and these are all part of the ER, is there some way we can make sense for what a single protein might experience based on where it is inside the structure? And to try to address this question, we developed a technology which is based upon a very old tool developed in my postdoc advisor's lab now nearly 20 years ago, which is known as SPT-PALM. And this technology is actually in principle quite easy. What you do is you individually photoconvert single proteins. So they are off, dark, invisible until they are stochastically reactivated. And when that happens, you track their location by very high-speed microscopy. And the result of this is you generate something like these green trajectories that you see in the movie here on the right. These proteins, as they're moving around, though, and the resulting trajectories that we get are actually not very useful to us. This is the equivalent of tracking the GPS on your cell phone, but we don't have any way to put it back onto a map. I might be able to see that you drove to work or to school or something this morning and stopped, but I don't know what this thing is that you stopped at. So the idea we tried to implement was to basically address exactly this question by trying to put an underlying map. And this worked by simply combining two different microscopes, one that tracks single proteins and one that tracks the underlying structure in which those proteins are moving. So you can see here in this movie on the right that these proteins are moving around inside the endoplasmic reticulum. And it turns out once you unwrap the boundaries of this structure correctly, traditional biophysical tools like mean squared displacements or velocity autocorrelations suddenly start working again, which before you knew where the boundaries were, these things would give you erroneous answers. So we set out to look for a question that we thought this might be the right tool for. And the question we set out on was trying to understand something about this phenomena of interorganelle communication that happens at contact sites between organelles. So the fact that organelles are physically distinct entities with luminal compartments that don't directly talk to one another gives cells a challenge. They have to calibrate what's going on inside mitochondria, for instance, with what's going on far away inside of endosomes. And it seems that the way cells have decided to deal with this is by communicating through these sites of direct contact with the endoplasmic reticulum. So there's an interesting problem that comes from this, though. You can see in this movie from a previous paper from the lab where we're imaging 7 different organelle compartments in real time, that these contacts, some of them, like the ones you see close to the nucleus here, are quite stable. But there are others, like the ones you see out here at the edge, that seem to form and break very, very rapidly. And so we'd like to understand how something can be both specific, but also dynamically regulated. What you're looking at here is a single site of contact between the endoplasmic reticulum and a mitochondrion. The mitochondrial membrane is colored in blue, and the ER membrane is colored in green. This is from a 3D EM volume, so this is data, not a cartoon. Looking at this, you can hopefully see right away that there is a lot of alignment between things inside the structure. For instance, mitochondrial cristae all have one end docked on the endoplasmic reticulum at the interface. So this tells you there must be specific proteins there that control this interface. Now, fortunately, there's many years of biochemistry that have studied to tell us who these proteins might be, but visualizing these interactions has yet been elusive. To try to address this, we thought, okay, let's look at the ER localized tethers that bind to the mitochondria by single particle tracking PALM, so we can understand something about what these sites look like and how quickly proteins interact with these sites. So we've applied this exact tool I just explained to you to this question. And what you hopefully can see here in sort of our first batch of data is if we just show you the trajectories of a control, which is just a fluorescently labeled protein targeted to the ER membrane. That's the tail anchor shown in the top panels here. This protein diffuses freely through the membrane with no obvious places for interacting with anything. However, when you look at the canonical ER to mitochondrial tether, VAT-B, which is what we're using as a model protein here, you can see that it also diffuses through the ER membrane, but it also shows sites of specific contact or interaction. These are most clearly visible by performing probability mass function analysis, the details of which are probably not important, but tell you that these are the sites where the protein is most likely to be found. And you can see when you compare these two that the control is equally likely to be found anywhere in the structure, whereas the VAT-B molecule, it has these hot spots that we think are likely to be contact sites. To validate that these might be contact sites, we built an additional arm to the microscope and also imaged where the mitochondria are. So here you can see the endoplasmic reticulum in cyan, the mitochondria in blue, and the site of the tether interaction are these hot spots shown here. And hopefully what you can appreciate from this is that everywhere these two organelles touch each other, there is a hot spot, whereas the tethers rush to that site and try to stabilize the interaction. In order to preserve our sanity, one thing that we found that we were very happy to see is that we can also see hotspots for this protein that are not associated with mitochondria. And this would have been expected since this protein is known to also bind to other organelles. If we hadn't seen this, this would be worrying. One last control to convince you that what we're looking at is really binding. If we cut the binding domain of this protein off, you lose 100% of the contact sites. They're essentially completely gone. And if you fuse this binding domain non-specifically to a different protein in the ER membrane, you can see that you rebuild these contact sites, suggesting these are really interactions. However, these are more than a super resolution image of contact sites, although they are certainly that, because each of these images is made from individual proteins that we tracked before they went to the site, while they were bound at the site, and then after they left the site. And so you can plot the position of a single protein for here, or in this example, one single tether that diffuses through the ER, binds to the mitochondria, and then leaves back into the ER only to be recaptured by the mitochondrial contact site. And this really amazed us because this is a very fast interaction. So to try to understand something about this dynamic, I'm showing you this movie here where I have slowed this down. This is going 1/3 of the speed of real imaging because otherwise it's too hard for your eye to follow. I've gotten rid of the endoplasmic reticulum because it makes everything confusing in the image. So all you see here is the mitochondria trace, 3 different contact sites on this mitochondria, and the trajectories of single tether proteins diffusing in the ER and then occasionally becoming captured inside of the contact site before occasionally leaving that contact site and re-engaging with a separate contact site. What's amazing is if you look at many movies like this, you can actually quantify this time of interaction. And what we found, much to our surprise, is that the average time that a tether spends binding the ER to the mitochondria is somewhere in the range of 500 milliseconds. This is far, far faster than any of the conventional microscopy tools would have been able to tell us. So we found this to be a very interesting observation because we know that these interfaces are how cells make decisions about changing what's going on inside of the cell. And now we understand something about how they can make decisions very quickly because the individual components inside these contact sites are changing over essentially every 4 or 5 seconds. It turns out we actually have a little bit more information though, because each contact site in our data set has many tens or hundreds of individual molecules that diffuse into it, interact with another protein there, and then leave. And we can ask if there are contact sites that don't move during the movie, if they're stable, can we ask something about what the common trends of behavior are for tethers inside the contact site? And what we found, much to our surprise, was that essentially every contact site in all of our data sets had a similar structure which was a low diffusion well that corresponded to where the tether was most likely to be, and it gradually returned to the diffusive speed of the surrounding endoplasmic reticulum at the edges. And we decided that the most parsimonious way to interpret this data might be that as you move towards the center of a contact site, tethers are more likely to see their binding partner. And so you get a gradual increase in the frequency of binding close to the center. And if this is true, you might be able to predict that we would see something of that in the underlying electron microscopy structure. So it turns out that that is actually exactly what you see. So the endoplasmic reticulum does actually take on the shape of the surrounding mitochondria at that site. Just in the last few minutes of this talk, I'd like to just quickly talk about one interesting application of this technology that I think is really makes me feel very excited about the potential to use this technology for things that are medically relevant in the future. So you may be familiar with the fact that this protein we've been talking about, VAT-B, our model tether, actually has a single point mutation in it, the P56S mutation, which was originally identified in a family in Japan. And this mutant form of this protein causes a very severe and penetrant form of Lou Gehrig's disease, ALS, in humans. And this really debilitating disease is associated with increased ER to mitochondria communication phenotypes. But this is very confusing because the biochemistry experiments on this protein suggest that it is misfolded and does not bind the ER to the mitochondria very well. So to try to understand this, we decided to try to track the diseased forms of this protein. And what you're looking at in this image here is just a few example trajectories of the diseased forms of this protein in a human cell. And what you can see here is there are many of these, and actually I've removed most of them to make the image not confusing. The vast majority of these are indeed aggregated in the cytoplasm. That's what these black trajectories here that I've shown you are representing. However, there are also trajectories that are still diffusing throughout the structure and are able to pass sometimes into the contact sites. And the ones in the contact sites I've color-coded here in red. So you can see that they still show interactions. What's very odd about them, though, is that their interactions are different. Notice this example here on the left. This is a single ER mitochondria contact site with a single trajectory of a single tether. That is a normal, healthy tether. And you can see that it is going all the way through the entire contact site, exploring every bit of the interface. However, when you look at the diseased mutant form of this protein, you can see that it becomes trapped in just a small subdomain of the contact site. And we wondered if that might mean that it didn't exchange so quickly. So long story short, when you measure this over very many different contact sites, that's exactly what you see. Actually, the diseased form of this protein actually becomes trapped in these contact sites. Nearly 30% of the molecules never leave the contact site while we are imaging them, in contrast to the wild type ones that have a mean leaving time of just 500 milliseconds. So this really surprised us, but also I think gives us some hope that we might be able to understand something about why this protein causes elevated ER to mitochondria communication. It has removed the ability of the cell to effectively restructure or change out the components of these interfaces. So with that, I'll just conclude. I think ER biology largely occurs at the level of single proteins, and this will probably be true for essentially all membrane compartments in cells. And if we want to understand how proteins are interacting in their membrane-based environment, I think it's going to become incredibly important that we can see them one single event at a time, because nanoscale biology can be plastic, it can be rare, and it could be influenced by something that is not obvious when you are looking at many hundreds or thousands or tens of thousands of molecules that are averaged together. With that, I'll conclude. This is my postdoctoral advisor and her lab, Jennifer Lippincott-Schwartz, and all of a wonderful team of collaborators, and particularly Craig Blackstone, who's a clinician who helped us get the samples to understand the Lou Gehrig's form of this disease. And to physicists who I worked with extensively, Chris Calderon and Jean-Baptiste Masson, who helped us to develop the algorithms to understand this kind of data, which has really been a major part of the challenge. With that, I'm starting my own lab now in San Diego, and I hope some of you will at some point have the chance to come visit me and Itai there. Thank you so much.
Hey, thank you so much, Chris, for the fantastic talk. So we have a couple of minutes for questions. We can put them in the chat if for the audience, and I'll read them to Chris, or you can. So maybe, so we have one already in the chat, and maybe I'll just read that. That's okay. So the question is that Sec61, the protein, is used as a marker, which is also a component of the translocon. In your analysis, do you only see the rough ER, which is where translation is happening, or are there other regions for smooth ER?
In our experience looking in tissue culture cells, so I have not looked extensively in many different primary tissues, but in tissue culture cells, there's very little delineation between smooth and rough ER. And if you track single proteins inside the network, they essentially will explore the entire network. So the Sec61 beta molecules that we have tracked, I should provide a disclaimer that if you put a fluorescent tag on Sec61 beta, it no longer binds to the translocon. So it sort of becomes just a sampler for the membrane. It's no longer just marking sites of translation. But when you track it under those conditions, you can see that it explores the smooth and the rough ER quite efficiently. And we do see in the 3D EM, I'm not showing this data, but we do see ribosomes frequently docked out on tubules and other things. I don't think for tissue culture cells, at least in cancer cells, they maybe have lost the ability to really delineate these two subdomains.
Okay, so another question is, how did you define the area of the mito ER contact site where you show it surrounded by the dotted line when you overlap them, the trajectory of the molecules?
So the contact sites are actually identified from the places where we can see the molecules bind. The molecules go in and out very, very quickly compared to the lifetime of the contact sites. So basically, you are looking at the average of all the places we see binding during the 30-second window, and then the trajectory, the single trajectory you see maybe takes up, 1.2 or 1.3 seconds of that time, if that makes sense.
May I ask one question? Yeah, from Yasu. Yeah, I'm very surprised at seeing the single molecule can move very smoothly and fastly in the inside of the ER. I believe that, but the specific region of ER has a specific protein. So how how a specific protein can be tagged with a specific region?
So the only proteins by single particle tracking that I can see clearly choose unique regions, right?
I see.
Sometimes they will show averages somewhere, but most of them go everywhere, right? They just spend more time in a certain place. The only proteins that I think have clear delineations are ones that bind to cytoskeleton or bind to something else rigid. IP3 receptor is a good example, right? This is a very large complex that you can track in the membrane. And IP3 receptors will often be quite stable close to other organelles. And I presume that's because they're interacting with very large complexes on the other organelle as well. And so the drag of two things together in the membrane is quite high.
Okay, so it means if you show the movement of the single molecule you used in a different region, so you might see the different result. So like a no moving or something like that.
I have not seen anything that completely stops unless it is bound to cytoskeleton so far.
Okay.
And if you break the binding interface, then they start moving again.
I see. Thank you very much.
Thank you.
Okay, So we have a couple more questions in the chat, but maybe we're going to save them to the discussion at the end, if that's okay. So Yasu, if you could come back on to introduce Dr. Morigaki.
Okay. So finally, I can introduce Dr. Morigaki Kenichi. He is a professor of the Graduate School of Agricultural Science of Kobe University. Today, he is going to talk about micro-patterned model membrane platform for studying the membrane structures and physiochemical property and functions. So Kenichi, please.
Yeah, thank you very much for the very nice, kind introduction. I'm very pleased to have this opportunity to introduce our work to you. So I'm very grateful to Dr. Shirai and also LINK-J organization. So what we are trying to do is make a model system of the biological membrane. I'd like to start my talk by introducing you about cell membranes. As you are aware that cell membranes are made of lipid membrane, and also it has some associated proteins. So it's a supramolecular system, and it's involved in nearly all functions in the cells, including signal transduction and energy conversion, like photosynthesis. And what basically the membranes are doing is that it provides two-dimensional structure with two-dimensional fluidity of the molecules, so the molecules can move around. And it has certain lateral organization. And it also divides the space into a three-dimensional space into compartments. And it actually has very complex system like lipid domains or protein clusters or membrane clefts. And we believe that these supramolecular structure are very important for various functions, but it's still very difficult to study how these supramolecular structures are actually regulating the functions. So there are many approaches to tackle this problem. And one is studying in vivo using cells like the previous talk by Dr. Obama. And the other approach would be to in vitro using purified molecules. And nowadays, we have also opportunities to study in silico by simulations. And we are going a slightly different way of constructing model membranes in a bottom-up approach by putting different molecules together to form a model system of the biological membrane to learn how they are organized and how they work. And there are different types of model systems like lipid vesicles, black lipid membrane, BLM, and supported lipid bilayer. And we are especially focusing on this third type of supported planar lipid bilayer. We call it SLB because of its mechanical stability. And also it's the fact that the membrane is at the interface between the solid substrate and the aqueous, liquid aqueous solution gives it opportunity to study the membrane using interfacial analytical methods. And we can also apply microfabrication techniques like micropatterning, which we have already for various, for example, integrated circuits. And we can prepare this membrane very easily from lipid vesicles by the process called vesicle fusion. So lipid vesicles absorb on the solid surface, on the hydrophilic surface, and destabilize and rupture, and it forms a continuous membrane by the fusion of the bilayers. And as I said, you can also apply micropatterning technique to form different patches of the membranes. So the concept would be we want to study the biological membrane by utilizing self-assembly of the individual molecules. So the molecules have the tendency to assemble into very complex structures. But at the same time, we apply the microfabrication technique to regulate how they self-assemble so that we can kind of design the membrane systems. And finally, we will analyze the membrane very in detail so that we can get information and also we can utilize it for biomedical applications. So for that purpose, we are using this lithographically patterned lipid bilayer. So that I developed more than 20 years ago. So we are using this diacetylene-containing phospholipid, which is a typical phospholipid that forms biological membranes, but it forms a photopolymerizable moiety, diacetylene, and it can polymerize by illuminating UV light. So if you prepare lipid bilayer on the surface of glass or silicon as monomer and illuminate UV light, you can cross-link lipids so that you can form a very stable lipid bilayer structure. And at the same time, you can keep some region as a monomer by using a photomask that control the area of illumination. And then you can selectively remove this monomeric lipids by using detergent solution. And polymer remains because it's very stable. And in the final step, you can apply natural lipid bilayer, incorporate it, so that you can have a combined polymerized and natural lipid bilayers in an arbitrary pattern. And in this system, the polymer acts as a scaffold to pattern and stabilize the membranes, whereas reconstituted natural membranes, we call it fluid bilayer, acts as a model system of the biological membrane. And there are a number of polymerizable lipids that have been developed in 1980s to 1990s with the aim of stabilizing lipid vesicles for drug delivery purposes. And as I show some examples here, and we are using this diacetylene containing polymerizable lipids because we found out that it is most efficient to stabilize the membrane. And so when you polymerize the membrane, so this is an atomic force microscopy image of the polymerized bilayer. And this area is lipid-free, so we remove the monomer. And you see that there is a gap of the high difference, about 5 nanometer, which corresponds to the lipid bilayer thickness. So we can see that this area has a polymerized lipid bilayer structure. Then we reconstitute natural lipid bilayer by the method called vesicle fusion that I introduced. And here we have the very unique observation that preformed polymerized lipid bilayer actually catalyzes the reconstitution of the natural membrane because it exposes hydrophobic core of the lipid bilayer. So it acts as a catalyst to destabilize lipid vesicle and form a planar lipid membrane on the surface. And this process can be observed by total internal reflection microscopy. Here I show you the video. So here is the boundary between polymer and the glass surface, and natural lipid bilayer starts to form at the edge of the polymer bilayer and propagates to the center of the lipid-free region. So in this way, we can show that we have a hybrid membrane of polymerized and natural lipid bilayers. And the important thing is that two different types of bilayers are integrated as a single lipid membrane. And if you incorporate a natural membrane and apply an electric field, you see that negatively charged fluorescent molecules are accumulated in the positive side of the electric field, whereas it is confined. So this shows that the membrane is continuous, yet each membrane is confined in a polymerized lipid bilayer. And this also gives us a unique feature that you can not only have completely polymerized and completely natural membrane, but you can have a composite membrane of polymerized and fluid natural lipid bilayers by changing the UV dose for the polymerization. So when you have a lower UV dose for the polymerization, you have a lower coverage of the polymer, whereas if you have a higher UV dose, you can have a higher coverage of the polymer. And you can have different regions, completely polymerized and completely natural and partially polymerized barriers. So you can control the ratio of the polymerized and the natural lipid barriers. And this can also induce phase separation of different lipids. In the biological membrane, we believe that there are a lot of different lipid domains which contains cholesterol-enriched and cholesterol-depleted regions. And we can reproduce this kind of phase separation in a pattern manner if you have a mixture of two lipids and cholesterol. The cholesterol-depleted region accumulates in the partially polymerized region, whereas the cholesterol-enriched regions are accumulated in the polymer-free regions. Okay, so we can have patterned lipid microdomains which mimics lipid domains in the nature. Okay, so we are using this kind of pattern membrane for studying the effect of lipid raft of protein clusters. And I can show you some examples how we apply them. So one example of the application is to study the affinity of rhodopsin to lipid phases. Rhodopsin is a G-protein coupled receptor in our retina, and it is kind of prototypical GPCR studied, very relevant for drugs development. And we obtain rhodopsin from bifrog retina and reconstitute it in a pattern membrane which has a preformed liquid ordered and liquid disordered, so cholesterol enriched and cholesterol depleted regions. Then we observe individual rhodopsin molecules moving around in this pattern membrane. So this dark area is cholesterol enriched, and this red area is cholesterol depleted. And we can count the number of the molecules if they are more enriched in the cholesterol enriched region or cholesterol depleted regions. In this way, we can see that rhodopsin molecules at different stages, if they are activated or not, or if they are dimerized, how much they have the affinity to this cholesterol-enriched regions. So this is one example of how we can apply the pattern membrane to study some biological questions. Okay, and another application would be to study photosynthesis. Photosynthesis in the plant occurs in the thylakoid membrane in chloroplast. So it has a very high amount of different types of membrane-associated proteins, and it regulates photoenergy conversion to proton gradient or reduction. But how this energy transfer is regulated in the two-dimensional membrane structure is not fully understood. So we can reconstitute thylakoid membrane together with phospholipids to form a planar membrane like this, and we can incorporate various types of membrane proteins into the pattern membrane. And we confirm that this reconstituted membrane proteins has actually a native functionality, so that we are now trying to study how these two-dimensional organization of the proteins are actually regulating the photosynthesis. Okay, in this, the last example, how we can apply the pattern membrane is the actin nucleation of the membrane. So actin is regulating a lot of things in the cell, not only the shape, but it also regulates organella, as have shown in the previous presentation. And we can, this actin filament is actually nucleated on the surface of the membrane. So we can mimic this actin filament formation on the pattern membrane so that we can synthetically form actin filaments on the pattern membrane. So this movie shows that how actin filament, bundle of actin filaments grow on the surface of the pattern membrane. And we can now study in detail how these membrane organization actually affecting the actin nucleation and the formation. Okay, so these are something that I wanted to share about the two-dimensional structure of the membrane. And in the next couple of minutes, I'd like to share how we would like to study three-dimensional structure compartmentalization by using the pattern membrane. And we are aware that membrane clefts, as I have shown in the membrane contact size in the previous presentation, is very important. And we are having such tiny membrane cleft in various systems like cytochrome membrane in chloroplast or mitochondria, or endoplasmic reticulum, or cell-cell junction, or synapse. And people are now realizing that these nanometer size confinements are actually having quite different physical chemical property of the water, and also confined molecules like higher viscosity water or retarded diffusion of the water soluble molecules. So we are asking how physical chemical and functional effects of nanometer size confinement are affecting the membrane behaviors of the membrane proteins and their functions. But it is very difficult to study these non-confinements in the real biological system. So we wanted to reconstitute this kind of non-confinement by ourselves. So to this end, we are now functionalizing the surface of the polymerized lipid bilayer using a molecule called phosphodieethanolamine. So far, I have been using this phosphatidylcholine, which has a choline moiety on the surface, which is very suitable for forming bilayer structure, but it is chemically inert. And we have also in the nature phosphatidylethanolamine, which has a primary amine moiety, which is chemically reactive. This is less suitable for forming bilayers, but you can mix phosphatidylcholine and phosphatidylethanolamine to form a bilayer, and it functionalizes the surface. And we use this PE for functionalize it and for use it, for example, for cell attachment or attaching some micro or nanostructure made of PDMS, polydimethyl siloxane, which is very often used in biomedical applications. So this is an example of attaching cells by grafting a peptide having this RGD sequence, which is recognized by cell. You can selectively attach cell onto the surface of the polymer. So, and if you have a different peptide sequence, or if you have polyethylene glycol, or if you have only bilayer, you don't see any attachment of the cells. But we are more interested in attaching the three-dimensional structure of PDMS, as I have introduced. And this is an example of the attaching PDMS microwells, so which is a small wells made of in the size of like 50 micrometer, and attaching it with the membrane. So these PDMS and membrane do not usually have the affinity to each other. So it's not usually you cannot bind these two materials. But using this biotin stereotactic linkage on the surface, you can attach these two materials. And in this way, you can attach two-dimensional membrane with three-dimensional microstructures. But we also wanted to have a even thinner gap structure by using an adhesion layer with a controlled thickness. So we call it another compartment. So, and the idea would be by attaching the membrane and the PDMS, you can have a very thin gap structure, like several tens of nanometers. And if you have a mixture of molecules, those molecules that bind to the membrane can diffuse laterally, and they can be incorporated into this gap structure more efficiently. Whereas those molecules that do not bind to the membrane are mostly left here in the three-dimensional aqueous solution, and they do not effectively go into this thin space. And we can detect these molecules with a high signal to background ratio in this very thin gap structure. As an adhesion material, we have used different types of materials like lipid vesicles or silicon nanoparticles or polymer brush. And this is the way how we fabricate the gap structure. We first generate a pattern membrane having polymer and fluid bilayer. And then we attach the adhesion layer. So by selective binding with these materials. And then finally, we attach PDMS onto this adhesion layer. And we do it in an aqueous solution. At the beginning, we have a fairly thick aqueous solution, but with time, this PDMS goes down, and finally it touches down onto the adhesion layer, so that this part is attached, but this part doesn't have an adhesion layer, so you have a gap structure with an aqueous solution. And the thickness is basically controlled by the thickness of this adhesion layer. Okay, so this is an example of combining this with a microchannel and this nano gap junction. So as I have said, if you have a mixture of molecules, those molecules that specifically bind to the membrane can diffuse into the gap structure. And in this example, we have a mixture of choleatoxin subunit B, which can bind to the membrane, having some glycolipid on the surface. And then this CTB can penetrate in the gap structure. Whereas if you BSA, albumin from bovine serum, cannot bind to the membrane, so it stays in the this microchannel. And if you have two colors, you see these binding even in the microchannel. But if you have the same fluorescence for CTB and BSA, you have very high background noise from BSA in the microchannel. So you don't see the binding of the CTB to the membrane. Whereas in the non-gap junction, the CTB is practically eliminated. So you have a much lower background noise, and you can see the binding of CTB more specifically. And we also use the silicon nanoparticles, which has sizes of from 10 nanometers to 10 micrometer, and which has a very well-defined size. And we coated the surface, for example, with lipids. And we again use the biotin lipid to attach it to streptavidin, and we use it for the adhesion layer. And the advantage of this technique is that you can choose different sizes of silicon nanoparticles, so that you can change the gap thickness by changing the sizes of the silicon nanoparticles. So silicon nanoparticles having 30 nanometer size can form a gap structure with a thinner, smaller thickness. And also this leads to a higher signal to background ratio, because you can suppress the non-specific permeation of the molecules by having a thinner gap structure. So if you compare 100 nanometer and 30 nanometer, you see that 100 nanometer silicon nanoparticles have a higher background noise compared with signal. And with 30 nanometer, you can suppress the background fluorescence more efficiently. And we can also modify the surface of silicon nanoparticles using some polymer material that is very resistant to a non-specific absorption of the proteins, so that you can use this gap structure for studying membrane proteins. Okay, so we are now applying this technique for studying the biomedical applications, so detecting some biomarkers and also studying membrane proteins in the gap structure. And I show you quickly some recent results where you have this lipid membrane, and here you have the microchannel where you apply mixed solutions. And this lipid has an antigen, presenting antigen, and the antibody that can recognize to this the surface can selectively penetrate into this gap structure, whereas albumin molecules that cannot recognize surface stays in the microchannel. So in this way, we believe that we can develop a new diagnostic methodology using this detecting molecules in the gap structure. And another application is reconstitution of the membrane proteins. So we reconstitute typically membrane proteins in the from solubilized stage, and those molecules that were selectively reconstituted can diffuse in the membrane and go into the gap structure. But whereas those molecules that could not be reconstituted stay mostly in the inlet of the inlet. So as a result, in the inlet, you see a lot of background fluorescence arising from aggregation or not reconstituted molecules, whereas in the non-gap junction, you see only molecules that could be selectively reconstituted. So we are hoping that we can use this non-gap junction for studying the functions and properties of membrane proteins at a single molecule level. Okay, so I have shared that we can mimic the two-dimensional organization, three-dimensional compartment using this pattern membrane and reconstituted proteins and non-fluidic systems. And this should lead to a quantitative membrane biophysics and biochemistry and physiology and biomedical and material applications. So as a summary, we can form lithographically polymer lipid bilayer combined with natural lipid membrane and nanometer size domains of polymeric and fluid barriers can be induced pattern space separation, and the membrane protein can be reconstituted, both purified and in crude membranes. And the polymeric barrier can be functionalized to attach molecule cells and 3D structures. And also, finally, I have shown that the nanometer size gap structures can be constructed that would be mimic in the future various membrane clefts like synapses or membrane contact sites. Okay, so as such, I'd like to finish my talk by acknowledging the people that helped me to realize this, and I am very happy to take any questions you might have. Okay, thank you very much for your kind attention.
Thank you, Kenichi. So you showed many possibilities that this technique can be used for the translation of the basic research to the medical and how do I say biophysiological research, I think. Do you have any questions, audience and someone?
I have a question.
Yes, please.
Yeah, beautiful talk, Kenichi. It's amazing systems, and there's so much interface with some of the biological concepts. I had a question about if you thought about trying to generate asymmetric membranes supported bilayer where the composition of the two leaflets is different, because that's a major topic now in membrane biophysics, and
So far, we haven't tried that, but some people, we know that some people are doing it by first making a symmetric membrane and then exchanging some lipids from the outer leaflet. Yeah, so practically you can do it, but yes, it should be doable.
Maybe I can add one more question then. In the last, you mentioned membrane contact sites at the end. So, that's what you showed with the nanoparticles, and you can control the distance really within the range in which these contact sites that Dr. Obara talked about, are acting at between 10 and 100 nanometers is really that zone. So to me, there might be a huge possibility to ask some fundamental questions, for example, about how lipids get trafficked and the distance dependence of that process using your systems.
Yeah, I think we can make something like 30, 40 nanometer gap structure right now. But as you can imagine, it is more technically challenging to have a thinner gap structure. But theoretically, it's possible. Yeah, so we are working on that.
Thank you. Can you unshare your slides?
Yeah. Okay. Can you see my slides?
Still, we can see. Okay. I say, okay. Okay, Itai. So we have 2 or 3 questions for Dr. Christopher Obara. Can you see the question?
Do you want me to take over?
Yeah.
Okay, yeah, we have some questions in the chat, I think, for both speakers. So let's just start. So Chris, there's a question about the P56S mutation, the ALS-associated mutation. What does that cause for the ER functions? What can the ER not do with that mutation on BAP-B? Is the lipid composition of the ER affected?
Great question. Unfortunately, I don't think I have good answers for you for any of this. There's been very little bit of work now. I know there are people currently working on trying to do lipidomics on these neurons, but as you can imagine, they're quite precious, right? The way they do it is they get stem cells from patients and then have to differentiate them in a culture dish, which is a relatively expensive protocol. So to my knowledge, we don't have like good lipidomics on this system yet. I think that would be really cool. The question about what happens to the ER, I would guess that VAP-B's function is primarily through the ability of the ER to coordinate and regulate the other compartments, which is probably the reason that we see so much associated with ALS-induced disease is lysosomal dysfunction and mitochondrial dysfunction, because the major way those two compartments speak to one another is through contact with the ER. So if what you're doing is disturbing the ability of the ER to regulate those interfaces, I think you disturb the ability to regulate what they're doing. And that's where I think everything goes awry. But I'm really out on a limb here. We don't have enough data to show any of that at this point. This will be years of work, I think.
Okay, next question, maybe we'll go back and forth. So Kenichi, there's a question about the maximum pressure tolerance of the new PDMS structures.
Okay, so the PDMS itself can be quite thick, so several millimeters. So it is fairly tolerant to the pressure. And maybe the more weaker point would be the attachment of the PDMS and the membrane. And we are working on to improve the glue chemically, so to enhance the stability. But I cannot tell you what is the exact pressure that we can work on at the moment. Yeah, thank you.
And then, okay, another question. This is for Chris. That's a general question. Can you comment on the use of AlphaFold or AI in ER-related problems?
I think there's a lot of people working on that right now. One of the difficulties with AlphaFold, I'm sure, as you're all aware, is that it has to train on the data sets that we have. And so I think AlphaFold works really well for ER proteins that we have model structures for. Remember, though, there's a substantial fraction of the ER proteome, which the membrane anchoring domains have never been solved. So all of the hairpins, for instance, we don't have structures for, and lots of these unusual twisted domains that things like Luna Park use or are thought to use to hold the ER structure in place. So we don't have any real thing to train the AlphaFold off of for that. So when you try to put those things in AlphaFold, they come out as noise. And so one of the things that our lab is working on that I didn't talk about today, we also do cryogenic microscopy, where we high pressure freeze cells, do fluorescence imaging while it's still frozen, and then use that to guide cryo-EM tomography, and then in situ protein structure identification. And one of the things we're hoping is that if we can take things that we know what their structure is, that AlphaFold can predict, and anchor them on the outside of the ER, we can localize where those things are, find those structures, and then use the associated scattered electron density to solve the structures of the domains we don't know. But this is like very much experimental technology right now, so I don't know if that will work or not.
Maybe since we're in kind of the discussion point, maybe we'll ask a question that kind of interfaces between the talks. So, I think, Chris, you're really looking in cells and trying to understand the behavior of the membranes and the proteins on the membranes within cells. And Kenichi, you're working in synthetic systems, more and more complex synthetic models, and maybe there's a possibility to bridge this gap. And I was wondering, has anyone ever reconstituted some of these tethers in a liposome, type system? And then could that be the basis for trying to connect it to the interesting models Kenichi has that might be relevant here?
I think maybe I will start to answers. But we would be very happy to collaborate with any type of biological questions. If you have a concrete question and if you have concrete molecules in mind, we have to have certain purified molecules. That's maybe one limitation. But if you have very concrete molecules and questions in mind, we can generate almost any type of structure. So it's not limited. So we would have very fruitful collaboration, like the case of actin filament. So if you biologists have some concrete questions that might be addressed by the model system.
Cool. Yeah, I think we're also really excited to see how these technologies can be implemented on top of other things. And, can you inform things from one technology that will guide the others? The reason these technologies came to be, right, is because we combined a few different things that people had already designed. None of this is like a new idea for microscopy, right? It's a combination of existing tools. And I think that's going to be the future for us going forward. So yeah, I think we're all very excited to participate in this kind of interaction.
And even the single particle tracking that you presented on, Chris, it has its origins also in supported planar systems. And Kenichi even showed single particle data in his talk. So you really see the interfacing between these approaches and techniques. Really, it was a really nice fusion of your two talks together today.
Yeah. Not only your two speakers, but also the audience enjoyed this symposium and the presentation. I think I'm afraid it's almost a time to close this session. Shinichi, can you take over?
Okay. Can you hear me?
Yeah.
Okay, great. And I'd like to say a few words before we close. Thank you very much for joining us for today's webinar. I'd also like to thank all the speakers and the moderators for their excellent presentation and discussion. I hope the audience found it both enjoyable and insightful. So to the audience, we value your feedback. So please complete the survey and let us know what topics and speakers you'd like to hear more about in the future. Your input is essential in making this seminar even more informative. So thank you once again, and we look forward to seeing you at our next event. And Waga-san, please.
Thank you. Wonderful talks and of course wonderful translations as well. And because Kobe University and UC San Diego will continue to collaborate on in this field. So those who are in Japan, feel free to reach out to Kobe University professors, including Shirai Sensei, and identify what might be relevant to your research. And also, eventually, that connection will lead to us in San Diego. So that will be a wonderful opportunity for everybody. I really, truly looking forward to people reach out to us. Thank you. So with that.
Okay, thank you very much.
Thank you. Take care.
Thank you very much. Goodbye now.